think I'm going to sort of really touch on some things that are not quite known to most people with regards to protein digestion. All right, so I've just sort of given you a schematic diagram of the digestive tract of a carnivore over here. I mean, it's very similar to most monogastrics. You have an esophagus, goes into the stomach. Then you have the blue, which is the small intestine, the green, the colon. And then you have attached to that the liver with a common bile duct that's shared with the pancreas. All right, and the pancreas produces a number of enzymes, amylases, lipases, trypsins, a few other things. Right, together with the bile itself, which we discussed was important for fat digestion. In the stomach itself, most monogastrics produce hydrochloric acid, right, and they can produce an enormous amount of that. Humans produce about 1.5 liters of hydrochloric acid per day. Okay, healthy human. That's an enormous amount. Okay, hydrochloric acid obviously is a potentially damaging substance. So it's very, very fascinating how the stomach forms the hydrochloric acid. Hydrogen ions, chloride ions separately, pushes them out into the lumen of the stomach, where they then combine to form hydrochloric acid. And then how it forms a protective layer for itself in terms of a mucus layer that protects the stomach wall, obviously, from autodigestion. Obviously, the hydrochloric acid can't be produced inside the stomach cells themselves, because then it would actually cause a massive change in pH inside the cell. And pH is regulated inside the body, you know, very sort of neutral pH for normal function. So that is really, really important. But very important to that is the combination between hydrochloric acid and an enzyme called pepsin. Pepsin comes from pepsinogen, and then it's the main enzyme for protein digestion, and it occurs primarily in the stomach. All right. The whole system of hydrochloric acid production is fascinating. There's a hormone called gastrin. So when you have very low stomach acid production, all right, you're going to increase your gastrin production, the hormone, and the gastrin is going to then feed back to the parietal cells in the stomach, which produce the stomach acid, and they're going to increase stomach acid production. All right. But we sit with certain situations where we just cannot produce enough stomach acid, or if you take chronic medication to reduce stomach acid production, the proton pump inhibitors, which are now, I mean, one of the most common drugs sold worldwide for humans, right? Big money maker for some of big pharma, all right? But they will suppress your stomach acid completely, and you would end up with a very high gastrin level in your blood, okay? Because the gastrin is trying to tell you to make more and more stomach acid. So we're going to talk a little bit about the importance of all of those things now. All right, so just to show you the importance of the combination of hydrochloric acid and pepsin at the same time. Pepsin as an enzyme functions optimally at a pH of around 2. So as soon as you increase the pH, all right, pepsin will actually become less and less active. By the time you get to a pH of 7, it's pretty much destroyed. It's very, very important that that happens, because otherwise any pepsin that goes down your intestine in your gut, right, will actually cause damage to the proteins in your actual gut itself. All right, so it's a natural way of basically in the stomach, keeping the function of pepsin in the stomach and then in part of the small intestine, but by the time you get to the colon, pepsin's completely destroyed and non-functional anymore. And this is just to show you, there's a piece of tissue over there in hydrochloric acid. You can see that it's still whole, where there's no pepsin. If you just have pepsin on its own, you get some degree of digestion, but if you add the two, pepsin and hydrochloric acid, okay, then you get maximum digestion. All right, and this is really important. If you can imagine sort of the hyenas that are scavenging, eating a large amount of bone. You can't eat a lot of bones and then end up with a situation where those bones are going to get stuck in the intestine because they're not properly digested. You've probably seen many, many cases where bone gets stuck in a dog's intestine, and then you end up having to go in. Now that would eliminate that animal in the wild very, very quickly, and yet they can eat a large amount of bones, and within a day or two those bones are turned into complete rubber, all right, and gelatine type consistency, and that goes down the gut, gets absorbed, you know, and used as nutrients. So that only can happen with a very, very high production of hydrochloric acid. The hydrochloric acid will denature the proteins that arrive in the stomach. It will decalcify the actual bones as well, and then it allow pepsin to actually act on those proteins to break them up into smaller peptides and then ultimately in amino acids, and amino acids can then be absorbed. Very, very important here. I mean, and many veterinarians, I find when I talk to them, they'll talk about protein digestion and they focus again on the pancreas. So when you have a German Shepherd with a maldigestion syndrome, they will again feed them pancreatic enzymes, but they forget to some extent that the primary protein digestion is taking place in the stomach. So there are some enzymes like trypsins and peptidases produced by the pancreas, but they are nowhere near as important as pepsin in the stomach. Protein digestion, as I said, primarily takes place in the stomach, not due to pancreas. The pancreatic enzymes are completely secondary to that. All right. And that's quite interesting because in human medicine, you know, if you've got chronic problems with gastric reflux, they'll put you on these proton pump inhibitors which wipe out your entire hydrochloric acid production completely, okay? And they'll put you on those drugs for years. Right? And then they will argue that it doesn't have any real effect, you know, on you long term. But that basically means you cannot digest your proteins properly. You're relying then entirely on your pancreatic enzymes to digest protein, and that's going to be pretty inefficient. Okay. So, and that has major health implications, and I think we're only just starting to realize that right now, not only with regards to the absorption of certain nutrients like calcium and magnesium and some of the B vitamins, which are dependent on a very low pH, but also just in terms of what is happening to those that chronic maldigestion, which you're effectively giving yourself if you put yourself on those proton pump inhibitors. Okay, so we can focus on humans just for a moment, again, just to illustrate how kind of common thinking is not so clever. All right, so one of the things that people mostly don't realize is that when you have heartburn, when you have chronic gastric reflux, okay, the problem is not that you are producing too much stomach acid, the problem is that you are producing too little, all right? The issue is that the distal esophageal sphincter is mainly controlled by the gastrin content in the bloodstream. Okay, it's one of the main triggers for keeping that sphincter closed. All right, and you want to keep that sphincter closed because you don't want the acid to rise up into the esophagus. Now, when you produce too much gastrin, then it will cause relaxation of that sphincter. Okay, and then even if the stomach pH is 4 or 5, okay, that's still acidic. It's still going to burn your esophagus. Okay, and that is effectively what happens when you have low stomach acid production. Your gastrin level goes up, you get relaxation of the actual distal esophageal sphincter, and then you've got another problem. Okay, stomach acid, besides the digestion of proteins, a very, very important function that it has is the actual sterilization of the small intestine. Okay, most bacteria do not survive at a pH of 2. Okay, they will be destroyed. So it has. You can eat salmonella and E. coli and all kinds of things in the food as a hyena, okay, but because you're producing so much stomach acid and you've got a pH of 1.5, none of that stuff survives, okay, and that's why they can scavenge, right? But more importantly than that is the fact that it controls the amount of bacteria in your small intestine. Now, if you follow the small intestine, it has a very low number of bacteria. There are still bacteria there, okay, that function, and some of the lactobacillus and things that you would get, you know, from probiotics, that's where they're actually going to function, in that small intestine, not in the colon. Okay, by the time you get to the colon, the pH has risen to about 7 or 8, and you've got billions of bacteria, more bacteria than the cells in your body. All right, but it's very, very important in the small intestine that you don't have a lot of bacteria. And when you do have too many bacteria in the small intestine, we have what we call a small intestinal bacterial overgrowth. Those bacteria cause inflammation in the small intestine, right? And they start breaking up nutrients that you should be having access to. They will utilize those nutrients. And especially if you've got a clostridial inflammation or overgrowth in the small intestine, you're going to have a major, major problem. Right. The other problem with those bacteria is that they will start fermenting different nutrients. And if anybody knows anything about fermentation, it produces gas. Okay, so gas that comes from the small intestine it can go either way, up into the stomach, or it can go down into the intestine towards the colon. Problem is, when it comes back up into the stomach, it starts to push the stomach acid up, okay, and then eventually pushing it up into the esophagus as well. So you've got these two problems. The one, the sphincter not functioning properly. Two, the fact that you've got a lot of bacteria causing fermentation and about gas pushing your gastric acid up into the esophagus. Right? And both of those will then contribute to chronic heartburn. Now, sometimes people will also get a hernia, a esophageal or distal sphincter hernia. And that may have many causes, but usually it's people that are relatively overweight and not exercising a great deal, probably got to do a lot with the actual strength of the diaphragm. And if they're not exercising, the diaphragm is not exercised, so that becomes a bit lax, and then you get this chronic gas pushing up into the esophagus, and eventually it herniates out, and then you've got a chronic hernia and needs surgery in order to try and correct this chronic problem with reflux. All right, so the problem is that many people are treated with antacids. You take, there are various different brands of antacids, Rennies and whatever you take, and that actually, it helps with the symptoms. And even the proton pump inhibitors that stop the acid production, they'll stop the symptoms, because now your pH has gone up to 7. Right now you can reflux as much as you want to. It's not going to actually cause any inflammation in the distal esophagus, so your symptoms are taken away. Right? And that's what the big problem is when you have medicine that is focused on treating symptoms rather than treating underlying causes of the problem. Okay. So if you go and look in the literature, and I've now looked at scientific literature to try and figure out, okay, so what causes people to have low acid production in the stomach? It's a very interesting question, and I don't think that anybody really has a good answer for that. It's not something that's been studied in the literature. If you go to things like so Helicobacter, which is a bacteria was thought of to be the main cause of peptic ulcers, certainly is in humans a problem. But for me, they sort of market Helicobacter as this acid-loving organism that lives in your stomach. Well, it actually doesn't like acid at all. It hides in the mucus layer in the crypts of the stomach, away from the acid. When you drop your acid production, however, then Helicobacter can take over. It can then expand in the stomach, and that's when it becomes a major problem, right, and causes the peptic ulcers and so on. We look at third world countries that have a very low protein intake. The Helicobacter is a big problem in those countries compared to third world countries where we have much higher protein intake. So there may be something there in terms of the quality of the protein that you're getting in the diet that may interact. But I can guarantee you that you'll find very little published in the literature as to older people. As you age, your acid level goes down over time. So why is that? What aspects? Again, it's very difficult to figure out what the causes of that are. Okay, I've got some theories which we are going to talk a little bit about when it comes to cheetahs. They give us, that's what I love about studying different animals. You start getting the extremes, and then they give you some clues, I think, as to what what might be going on, in humans, for example. And so when you study multiple species, I think you get a much better indication of how physiology works by looking at these really weird and extreme situations compared to us who might have a sort of middle of the range kind of acid production. Okay. So maybe change the way we think about it. But when you're not producing enough stomach acid, you have a condition which is called hypochlorhydria. Okay, so basically with a pH at peak level when you're supposed to be digesting food is above 2.5. And the effects are quite profound. I'm just going to pop these all up quickly and then we'll go through them. All right, so we have a reduced bactericidal effect. So that means that your increased risk of pathogenic bacterial infections, Clostridium difficile infections in humans, particularly a problem on people that have been on these proton pump inhibitors for long periods of time. So increased risk of bacterial gastritis, and the question mark over Helicobacter, also things like Salmonella. You get an excessive gastrin production, which because of the lack of negative feedback, bacterial overgrowth of the small intestine, the proteins are not denatured and poorly digested. You get bacterial fermentation of the amino acids in the small intestine, and this is now an interesting subject because it's only, I think in humans, they've only recognized this in the last 2 or 3 years. Those are all the amino acids in the diet that have this phenolic ring on them, tryptophan, tyrosine, and phenylalanine. If they get fermented by bacteria in the gut, they produce actual compounds that have biological activity in the body. They are absorbed by the small intestine into the bloodstream, and they have effects on the brain and on the kidneys, elsewhere in the body. It's like taking a drug, but these are drugs produced by bacteria in your small intestine. And they are not inert. They actually do stuff in the body. And this whole gut-brain axis that people talk about. This is one of the reasons why you have that gut. It's not about what you've got necessarily in your colon. It's what you've got in your small intestine that is really important, and how effective your digestive efficiency is. Now, we'll just come back to taking like probiotics, for example. The biggest thing that affects the gut microbiome, the bacteria in your gut, are the two things that are not as to what you're exposed to or whether you take probiotics or not. The first thing is your digestive efficiency, okay, and secondly is the type of diet that you eat. Right, those are the two things that will most affect your gastrointestinal microbiome. Digestive efficiency is probably the most important, because bacteria in your gut feed on something. They feed on whatever you're putting in there. Now, normally what we do is certainly as carnivore, it starves the bacteria. They have very few bacteria compared to a herbivore, which actually needs these bacteria and microbes to be able to produce a lot of the protein that they actually utilize. But in a carnivore, because they get such a high protein diet, they actually really want to try and keep the bacteria in the colon starved. They get very little. But when you don't have proper digestive efficiency, then there's like a party in the intestine, because there's so much food coming in. You get Clostridial bacteria having a party, and they overgrow and they start producing toxins and fermentation, resulting in a whole bunch of drugs essentially that get absorbed into the bloodstream and go to various parts in the body and do stuff, which can be positive or negative. In most cases, negative. Okay, we discussed a little bit about that in terms of renal failure in cats. All of our cat species, I think, are largely due to the drugs that are produced by bacterial gut bacteria, not per se the amount of protein in the diet itself. Okay, so digestive efficiency really important, and then whether you have a high carbohydrate, high fat, or high protein, or mixed diet, that will affect the bacteria in there. One thing that we do know in humans is that the more carbohydrates you eat, a very high carbohydrate, the carbohydrates actually dilute the stomach acid to some extent. They act as a buffer. It acts as a buffer in the stomach, and therefore, if you combine high carbohydrates with protein, your digestive efficiency on the proteins is much less efficient. Okay, because of the buffering effect. So ideally, if you wanted to, you should eat the steak first, digest that first, and then have the chips afterwards, if you wanted to do it properly. So separating them to some extent does help your digestive efficiency. One stage, I don't know if it was a popular year, but in South Africa we had this food combining diet where you separated your carbohydrates from your proteins and fats. And I think the main reason why that worked to some extent was for exactly that. Got better digestive efficiency. Okay, and then the other things were the reduced absorption. So, older people that develop osteoporosis and end up with fractures and stuff like that may also be linked to some extent, besides the hormonal changes, but it may also be linked to the ability to be able to absorb things like calcium. You need a fairly low pH to be able to absorb calcium more efficiently. Magnesium, iron, zinc, also the absorption of those is also affected by gastric pH. And then the B12, vitamin K, and folate absorption are also affected. They need a very low pH in order to be properly absorbed from the gut. So I just want to tell you a little bit about these parietal cells, because they're quite amazing little things in the body. So you have these in the stomach, these big crypts, okay, and in them the variety of different cells that do mucus producing cells and so on. But these are the parietal cells. Now these cells are the main ones that produce the stomach acid. Now, they're really important because they, as I said, they've got these proton pumps. The proton pumps pump protons, like hydrogen ions, into the actual space, into the lumen. They also produce the chlorides, and on the other side, they pump bicarbonate into the actual bloodstream. Right, so very, very active, need a lot of energy to be able to do that. So in humans, we have these little things inside the cell called mitochondria. They are mainly there for energy production. And this is now an electron micrograph of a parietal cell in a human, and if those little black things that you see in there are mitochondria. Now, if you compare the total cell volume of this cell, compare that to the mitochondrial volume, okay, then in parietal cells we see mitochondria making up the biggest proportion of cell volume of any other cell in the body, right? So indicating that they are probably the most energy demanding cells in the body. And it's quite, even your muscles, your heart muscle, which is a very hard working part of the body, even your brain, which consumes an enormous amount of glucose, all right, is doing nothing compared to the parietal cells in the stomach in terms of energy requirements. And this becomes quite important in understanding, when we get to the cheetah, which I'm going to explain in terms of energy production and dynamics in cheetah. All right, so in humans, maybe mitochondria play a really important role in, as I said, the energy production there. Now, energy is quite important. You've got on both sides of this parietal cell, you've got to pump bicarbonate ions into the bloodstream on this side, you've got to put hydrogen and chloride ions on this side, right? So you've got the opposite ends, and somewhere in the middle you've got to get energy, ATP, which is the currency of energy currency of the cell, from one side to the other. All right, so the whole point of glucose or the oxidation of fats in the mitochondria is to produce that little molecule called ATP. ATP is used for energy everywhere in the body, but the important thing about it is you can't store ATP. You can't keep it for very long. You need to either store it in glucose, in fats, or in creatine, creatine phosphate. All right, those are different molecules, or different ways of storing ATP, and you get ATP out of that, and you use that for energy to drive those proton pumps. Now, one of the things that we're interested in cheetahs, and that I'm going to talk a little bit about, is creatine. I mean, you might know it as a supplement, particularly if you've been involved in some sort of sport, or bodybuilders use it. Creatine monohydrate, they take it as a supplement. And the reason they take it as a supplement is it increases muscular performance. Now, in humans, we actually produce creatine in our body ourselves, right? And if you're a 100-meter sprinter, human, you use creatine phosphate. It can store small amounts of ATP as energy, and if you had to do a 100-meter sprint, you would probably, the first 2 seconds of your sprint, you'll be using creatine, and that's complete anaerobic energy production. You don't need oxygen to produce that, to release the ATP to make it available, but you've only got it for 2 seconds. And then your 3rd second of the run, you have to start using glucose, breaking down glucose to provide the ATP for the rest of the 100-meter sprint. Now, the interesting thing is that a human athlete will produce or the breakdown product of creatine, we're going to discuss that in a moment, is creatinine, which you basically get rid of in your urine. A human athlete, high-performance athlete, will probably get up to a maximum of about 2 grams of creatinine per day that they'll excrete. Now, I'll show you the cheetahs going up to about 20 grams and it's an animal that weighs half the size of a high-performance human athlete. So they're using this creatine in a special way, and I'm going to try and talk a little bit about that and why that's important. Okay, so one of the important things about creatine is not only the fact that it can store a bit of energy, but it can move energy across the cell. If you were to just have ATP itself with no creatine, it would take 100 times longer for that creatine to move across from one side of the body or the cell to the other side of the cell. So creatine acts as a shuttle, moving the energy more efficiently across the cell. It's important, obviously, in skeletal muscle. We know that it's also important in the brain. If you have a person who is born with an inability to be able to manufacture creatine in themselves, and they don't get sufficient creatine in the diet, they actually become very mentally retarded. So they have real problems with learning and can be, quite impaired, mentally impaired. It's important, obviously, in heart function because it's a very hard-working muscle in the body. But then, quite interestingly, in the eye and ear, they found in mice that when you supplement them with creatine, you can actually improve their hearing, because creatine is those little fibers in the ear that vibrate. Okay, they vibrate based on primarily creatine that provides the energy for those vibrations, and that enables you to hear. So you can improve your hearing with creatine supplementation. And then sperm they have no way of producing their own energy. They function almost primarily on the creatine reserve to provide the energy for them to be able to swim. Right, and then lastly, one that's not really been studied, as I mentioned, potential of creatine for gastric parietal cell function. It makes sense if this is a high energy demanding cell that it also functions to some extent on creatine, and particularly in the cheetah, as we'll discuss. Okay, so just a little bit here about creatine and creatinine and how do you produce it in the body. Sorry, this is detailed, but I'll get to the point of this in a minute too, and where it comes from and why it's so important in terms of nutrition. So, all right, so you can get creatine from meat. Creatine is present in meat. And if you calculate the total amount, you can get about 3 grams of it per day. For a human, that's more than enough. I mean, if you're eating about, but then you have to eat about 1.5 kilograms of steak like a cheetah does, all right, in order to be able to get about 3 grams of creatine. And that would then go to the muscle. But on the other hand, you can start in the kidney, you can produce creatine through a process using 3 amino acids. The first 2, arginine and glycine, are combined to form guanidino acetate, and that happens, very interestingly, only in the kidney in cats. It happens elsewhere in the body, in other species, but in the kidney, this is what happens. And then that guanidino acetate goes to the liver, and there it gets methylated, and eventually that forms creatine. So 3 amino acids: arginine, glycine, and methionine. Arginine and methionine are very, very abundant in meat. Okay, so no problem there. Okay, you've got enough. If you're eating a meat diet, you're going to be able to do this with no problem. Glycine, however, is only present really in high volumes in collagen. Right, every 3rd amino acid in collagen is glycine. So if you don't eat enough collagen and you're eating just muscle meat, you're not going to get enough glycine, so you won't really be able to make enough creatine. Okay. Right, so creatine is then changed to creatinine, and then it's excreted in the urine, and we can measure creatinine in the urine, and we can get a good idea of exactly what is happening in terms of creatine production elsewhere in the body. All right, so for my PhD, I was interested in creatinine because when we look at any molecule in the actual urine of an animal or a human, what we tend to do is we try and look at the amount of creatinine excretion, and we think in humans that it's supposed to be fairly constant, and therefore anything else that you excrete, whether it's sodium or potassium or any other molecule in the urine, because humans drink socially, I mean, if you're going to drink a whole glass of water and I measure the concentration of a metabolite in your urine, then I need to know to adjust the concentration because you're producing more urine than the person who's a little bit dehydrated, and the concentrations of the molecules are going to be different because of the amount of water you drank. So in humans, they'll normally express anything that you measure in the urine relative to the amount of creatinine in it. Okay, it's just a way in which they adapt it. Now, I think it's a bit of a problem because I think they assume that it's related to muscle mass in humans, and I've shown in the cheetahs it's completely not related at all to muscle mass. Anyway, so we measure creatinine in the urine, and those plotted over there, you can see the blue dots are the females, the red squares are the males in terms of concentration, and then the age. And you can see there's a bit of a pattern, all right? So about 5-6 years of age, they reach a peak, and then it tapers off as these cheetahs get older. Now, anyone looking at that would normally say, oh yeah, if it's related to muscle mass, then as the cheetahs are getting older, they're losing a bit of muscle mass, and that's why their creatinine levels are going down. Well, the interesting thing is that there are at least 3 females there that are producing more creatinine than any of the males in their urine. And we know, I mean, certainly in Namibia, cheetah females are quite a bit smaller. They have less muscle mass than the males. So that immediately says, no, this is probably not true. And then when I looked at the body mass indexes of older cheetahs, these older cheetahs, there's no correlation. The older cheetahs were not losing muscle mass, and yet they're producing less and less creatinine as they were getting older. This little graph over here just shows you what happens in humans. Again, very similar sort of pattern, right? You reach a peak at about 20, 25 years of age, and then after that there's a decline in creatinine excretion in the urine. All right, so creatinine, very interesting. So when we want to, okay, let me just get the other 2 slides up here. When we wanted to catch black-footed cats in the Karoo, the black-footed cats, our smallest little cat species in South Africa, it weighs 1.5 kilos, a very tiny little cat with very short legs. All right, the little one up here. The way we catch them, they don't go into traps. So you can trap hundreds and thousands of hours, and you can put all kinds of things into the trap. They will never go into the traps. I mean, you get maybe one every year. That'll be it. So the best way to catch them, we travel at night on the back of a vehicle like this with a spotlight. You find a black-footed cat. You can recognize their eyes in the headlights, and then you chase them. All right. And in the Karoo, fortunately, it's a very flat area. Okay, occasionally have a few problems with a aardvark hole or something like that, which you really don't want to hit at high speed. But you can chase these cats, and 60 kilometers an hour. And I think they run almost faster than cheetahs do relative to their size. I mean, 60 kilometers an hour they're going, but they only run 500 meters. And for the females, maybe the males will run about 800 meters. So as long as you can keep up with them and you get to that stage, they'll stop running. Then they go and sit down. And then you sort of circle around them. Then you get off the back of the vehicle with a net and you go and put the net over them. You walk up to them and you hand inject them. And they just sit there. So one night while I was out, we saw a caracal in the headlight. Now, you know, caracal is a substantially bigger cat. And I thought, okay, let's see, can we catch a caracal in the same way? We were sampling a whole bunch of felids at that stage and chased this female. She ran about 500 meters, went and sat down, and I thought, okay, well, this is going to be interesting. Put the net over her, and I thought, well, I've never hand injected a caracal. I mean, they can be very aggressive. But I walked up to her, hand injected her. She went to sleep, collected the samples, woke up, no problem at all. That sort of got me thinking too. I mean, there's quite a lot of the questions around cheetahs. Why do cheetahs stop running? I mean, why do they only chase for such a short period of time and then stop running? And one of the questions was, well, don't they overheat? You know, isn't heat the limiting factor? So one of my colleagues did a study quite a few years ago now where they put these data loggers in, the temperature data loggers into the cheetahs at Africat in Namibia. And they monitored these cheetahs when they were hunting and when they were not hunting. And they found that when they were hunting, their temperature only went up by 1 degree. Okay, really didn't, you know, 1 degree going up from 38 to 39 is not going to affect your performance at all. All right. But the cheetahs will only run for maximum sort of 30 seconds, and then that's it. They're not going to run again. So I've got now an MSc student that's busy looking at muscle physiology in cheetahs, and they looked at the enzymes, different enzymes that are present in the muscles of cheetahs. And one of them, creatine kinase, which is the main enzyme that converts the phosphocreatine to creatine and allows for that regenerates it, allowing for that ATP to be freed, okay, is massively elevated in the cheetahs. So what it appears is happening, and this is probably true for all cat species, they are not endurance athletes. They're all high-speed, short-distance athletes. All right, they probably are running on using creatine as their primary energy source. But as I said, creatine in a human will last about 2 seconds into your run. In a cheetah, probably about 30 seconds. Okay, and it's probably true for, as I said, almost all the cat species. So it's very interesting because people have published quite a lot on cheetahs and why they can run so fast, looking at the anatomical, structure of their legs and back, the way in which the nose is structured. They probably don't even need oxygen when they run. They can run almost anaerobically for 30 seconds, probably because they're using mainly creatine for that energy. But when it's gone, then it's gone. Okay, and then you have to lie down probably all day to make enough, build it up in your muscles and in your liver to be able to then run again. All right, and that's probably why lions and all the cat species are so lazy. I mean, they lie around for most of the time and do absolutely nothing, and all they're doing is busy working on making more and more creatine, okay, so that they are able to run again. There's an interesting story I now read recently from the Indian Book of Mammals, published in 1847, where they also talked about the fact that the cheetah is actually very easy to hunt. You chase it down with a horse. And then you can just spear it, and probably one of the reasons why they were so easy to hunt. Although they can run away over a short distance, they're nowhere near like a wild dog or any of the canid species which can run and run and run and run and just not stop. Horses as well would use very different enzyme systems to be able to produce the energy for them to maintain a consistent speed over a long period of time, but they will never get up to the speed of the cheetah. So hopefully, if I have any aims at getting one paper into Nature, it will be this, proving why the cheetah can run so fast, not based on anatomy, but based on the biochemistry or physiology. It really hasn't been studied at all. But that comes down to, in order to be able to produce it, they need to get that one amino acid that I talked about, which was glycine, in adequate amounts. And as I said, glycine comes from collagen. If you look at these are the relative amino acids in collagen, glycine is by far the most, every third amino acid in collagen is glycine. It's the very simple amino acid. And cheetahs seem to be able to, this is again way too small, you won't see all of this in detail, but they seem to use this particular amino acid in bucket loads. All right, so not only do they use it to make creatine, but they also use it in many of the detoxifying mechanisms in the body. Whenever you metabolize drugs, there are two phases, phase 1, phase 2 metabolism in the body. Phase 2 metabolism is often where you join a molecule to some other, it can be sulfates, or it can be gluconates and so on, or glycine. Glycine conjugation, very, very important process to make the drug more water soluble so that you can actually urinate it out in your urine. Right? And when I did my PhD, these are a whole bunch of compounds which we found in the urine of cheetahs, and a large proportion of those with the arrows over there are glycine conjugates. So this is very unexpected because we didn't think that this was a major pathway in cats in general. But if we just calculate the amount of glycine that they use up here, you know, it already consumes a huge amount. Many of these, if you just look at the other compounds that we found in the urine, are metabolites of those phenolic amino acids. These are all tyrosine, phenylalanine compounds, a huge amount of them. So again, just proving that, and these are captive cheetahs, that they are actually fermenting those products and absorbing them, and then some of them are getting conjugated to glycine and being excreted in the urine. So just the point of all of that is just to show you that these animals need this particular amino acid that comes from collagen. Right? You know, you can find small amounts of glycine elsewhere, but you really are not going to find it very large amounts of that elsewhere. Now this is where it gets a little bit even more technical. Just this is that process that I was explaining to you about glycine conjugation, and it's you know, as I said, you metabolize some drugs in this way. If you take aspirin, a certain proportion of the aspirin is going to be excreted via glycine conjugation. If we take a phenolic compound like those amino acids that we talked about, this whole process of glycine conjugation takes place in 2 phases. And what is very interesting in the cats, again, in humans this happens in the liver and in the kidneys. Cats, it only happens in the actual kidneys. Again, one of those interesting things with regards to kidney failure in cats. It's a 2-stage process. First of all, the phenolic compound gets bound to coenzyme A. Coenzyme A, I mean, is like also very, very important, plays an important role in all kinds of different reactions in the body, mainly related to energy. All right. So now you have this compound bound to the coenzyme A. Now that forms what we call an acyl coenzyme thioester. Now normally that's not a problem because that although this molecule is highly reactive, it causes massive damage in the body if it gets the chance to hang around for very long. But fortunately, when you've got enough glycine available, phase 2 of that reaction, the actual conjugation to the glycine takes place. Glycine binds to that molecule and the coenzyme A gets freed. All right, and then you've got this glycine conjugate, which can just be peed. It's inert, and it will be peed out in the urine. All right, so no problem. But now you can imagine, if you don't have enough glycine available, you get a problem where that acyl coenzyme thioester gets stuck. It can't move. All right, that's 2 problems. The 2 problems with that is, one, we've already mentioned it's a very reactive compound. The second problem that you end up having is what we call sequestration of coenzyme A. That coenzyme A is supposed to be there very temporarily and then be available for some other reaction elsewhere in the body. Now when it's bound to this thioester molecule, that, you know, it's tied down, it's sequestered, it's in jail essentially, and it can't do anything else in the body. So we're going to end up with 2 problems. One is going to ongoing damage in the kidney. The second problem is going to be that coenzyme A, we're going to end up with a coenzyme A deficiency. And that's pretty much exactly what we see. Anyway, so we're coming back to glycine, this very important molecule in the body. It's the simplest amino acid. It's generally considered non-essential, because non-essential basically meaning that you can make it from other amino acids in the body, like serine and threonine and so on. So I put non-essential in question mark, commas there, because simply it basically means that questionable. Every 3rd amino acid we've mentioned, it's a very important inhibitory neurotransmitter. If you want to sleep at night, you can take supplementary glycine as well. There are 2 main neurotransmitters in the brain, GABA and glycine, that inhibit the function in the brain and allow you to sleep properly. All right, they just cause relaxation of the nervous system. And that relaxation of the nervous system is almost exactly the same relaxation that you get in the immune system as well. So they downregulate the immune system, trying to reduce inflammation by exactly the same mechanism, as they would with the nervous system. So you can see it plays an important role in just keeping that immune function, or keeping a lid on the immune function and preventing it from becoming a runaway inflammation. Lots of other things. So it's important in creatine synthesis, but it's also important in glutathione synthesis, which we spoke about earlier as an important antioxidant. Porphyrins, purines, those are important in the production of your DNA and so on. The production of bile acids, not so important in the cheetah, but in humans it's certainly important because cheetahs use taurine in the bile, production of bile rather than glycine, so sort of the only one way in which they kind of are helped. The conjugation of xenobiotics. Xenobiotics are those compounds produced by the bacteria in the gut that we talked about. Okay, so that conjugation process and any of the drugs as well. And then they modulate methylation. If you have a diet that's very high in meat, you're going to, muscle meat, you're going to produce a lot of methyl groups, and you're actually going to start over-methylating. And glycine is a way of balancing that out. So ideally, even as a human, if you eat a lot of meat, you need to eat a lot of collagen as well to kind of balance out the methylation reactions. If you over-methylate, you're going to turn on a whole bunch of switches in your body with regards to DNA synthesis and upregulate certain things and downregulate other things. So methylation reactions need to be balanced. I won't go into all of that detail. But so glycine, a critically important little amino acid in the body. As I said, you can make it from other things like threonine and serine, which are other amino acids. But there's been a recent paper, which I think I've put on here. Yeah. In which they did some calculations in humans to see just how much glycine we needed. And the conclusion of the calculations based on where you can get glycine from is that most humans would, may fall significantly short of the amount needed for all metabolic uses, including collagen synthesis, by about 10 grams per day for a 70 kilogram human. Okay, so even us as humans, we might end up with a glycine deficiency. Even if we can produce it ourselves from other amino acids, it may be a real problem for all of the reactions in the body. Okay, because we don't necessarily eat the same sort of diet that we would. You get very little glycine from plant material. So if you're on a vegetarian diet, that might be a little bit of a problem for you. Even if you are on a fairly high meat-based diet, that if you're not eating the whole carcass, if you're not eating all the collagen-rich tissues in your bone broth and everything else, you're not necessarily going to get enough glycine either. All right, so the potential consequences, and I'll finish with this and then we'll take a break, for cheetahs on not getting enough glycine is that you're going to have increased collagen turnover. The body will actually mobilize its own collagen in order to be able to provide enough glycine because it's such an important amino acid. So if you're not getting enough in the diet, you're potentially going to get an increased breakdown of collagen tissues in the body, bone and other collagen-rich tissues. Okay, so reduced bone strength. You're definitely going to have a reduced creatine production, which then has also going to reduce your bone strength, potentially reducing gastric acid production, okay, because you need that energy, and particularly in the cheetah. If the cheetah is using that creatine system in the muscles as a primary energy source, there's no reason why it would not replicate that in the parietal cells as the primary source of energy production in order to produce gastric acid. Why have two completely different systems when you can get away with one system and just replicate it from muscle physiology into the parietal cell physiology? Okay, you're going to have reduced sperm motility. That's going to reduce your reproductive potential in males. You're going to get potential neurological disorders, restlessness, anxiety. You're going to have reduced glutathione production. You're going to get reduced conjugation of your xenobiotics, which potentially increase your risk of renal failure. We'll discuss that a bit more in detail. I haven't spoken about this yet because it's a whole nother story. You're going to get reduced production of renal dopamine, and I will explain that a little bit later as well. Dopamine, very important neurotransmitter in the brain. Obviously, if you don't have enough, if your whole dopamine system is not functional, you get Parkinson's disease in humans. But what people don't realize is that there's about 20 times more dopamine in your gut and in your kidneys than there ever is in your brain. They play such an important role in regulating the gut function as well as in, and there are reasons as to why glycine deficiency would potentially cause a reduction in both renal and GIT dopamine production. And then chronic inflammation, because you don't have that suppressing effect, as I mentioned, with regards to the same effect that you would get in the nervous system. Sorry, and this was just, okay, I'm going to skip through that for now. Okay, let's just stop there and then I'll talk a little bit about that later. So hopefully, yeah, longer session. What are we now? Time was almost 12. I think lunch was around about 12. Okay, if I can carry on for 5-10 minutes, then we can just cover this last section. this was just to show you these aromatic amino acids, and part of my PhD I found all of these different breakdown products from these aromatic amino acids. Initially when I did this work, I thought, oh, this is what's happening in the colon. These bacteria in the colon are breaking this down, fermenting these different amino acids, these phenolic amino acids, and then these are getting absorbed into the gut and then ending up in the kidney, by coming out in the urine. But I've later realized that most of this is probably happening in the small intestine, and this fermentation is probably a real problem for these cheetahs, producing a lot of these things which are then consuming quite a lot of the glycine. And that's just the one for tyrosine. We've figured out where all of these things come from. Now, this is where the dopamine side of things comes in. So I found this obscure paper by this Chinese researcher, and he was looking at the potential inhibitory effects of tyrosine metabolites on a very strange enzyme called dihydropteridine reductase from human and sheep liver. Now, it's, as I said, quite an obscure paper, but they showed that there was quite significant suppression. So these things, these products of bacterial fermentation end up being absorbed, and this is one of those effects that I talked about in the body. And I found this to be the case. It made sense in what I found in the cheetahs. It definitely agreed with what I was finding. Now, just to quickly show you here, our hormones like serotonin, these are all the neurotransmitters, sorry, not hormones, in the brain, serotonin, dopamine, noradrenaline, adrenaline, they all come from those initial phenolic compounds, tyrosine or tryptophan. Tryptophan produces serotonin, tyrosine through a different pathway produces dopamine, and then from dopamine you get noradrenaline and adrenaline. Obviously these are really important in the body. Those are broken down by a group of enzymes called monoamine oxidases into these very strange sounding metabolites called homovanillic acid and vanillyl mandelic acid. They're not really of any importance, but one of our difficulties is we can't measure dopamine very easy in the body because it breaks down so quickly. So you can take a blood sample, and then you have to freeze it in liquid nitrogen, immediately take it to the lab, and then they will analyze it for you. Now that's quite a pain to do when you're in the field and you're trying to collect samples from cheetahs. But what you can do is you can measure the homovanillic acid and the vanillyl mandelic acid in the actual urine, and what they will do is they will give you a reflection of the total amount of dopamine that's being produced in the body, because they are direct breakdown products. So that's exactly what I did, was measured these two, and then I got a fairly good idea of what's happening. Now, just to point out that enzyme that we talked about in that previous study is involved in the recycling and the production of dopamine over here. So when I measured the differences in these vanillyl mandelic and homovanillic acid in captive and wild cheetahs, the wild cheetahs had quite a lot more, than the captive cheetahs. And for me, that was an indication that they were producing more dopamine. When I looked at the relationship between, this is a graph just plotting the relationship between those two, VMA, or vanillyl mandelic acid in blue, and the HVA in red, and then compared them to there were a whole bunch of these compounds that were glycine conjugates. This is phenyl propionyl glycine. All right, so it's one of those fermentation products that are produced by bacteria. You plot them, the concentrations in the urine, and you compare them to the concentrations of these other two breakdown products of dopamine. You can see a little bit of a pattern occurring there. And what I wanted to show you, so if you've got very low dopamine production, you'll have very high levels of those phenolic compounds in the urine. If you've got very high levels of these dopamine breakdown products, you have to have low levels of those products so there's some sort of relationship between the two. So when bacteria are producing these phenolic compounds, it appears that there's quite a good possibility they are suppressing the production of dopamine. So you've got this kind of inverse relationship, correlation between the two. And when you look at a whole bunch of them, you get a similar pattern in all of them. So very high concentrations of the phenolic compounds, breakdown products on the left-hand side, and as you get higher levels of homovanillic acid, you've got lower levels. So it's the same sort of pattern that you're seeing all along the way. So again indicating that there's a very good chance that those products themselves are inhibiting dopamine production. So dopamine in the GIT, we're going to finish with the next 2 slides. So dopamine in the GIT, what most people don't understand is that it's really important in gastric motility. It reduces the risk of gastric ulceration. So people with Parkinson's disease have a much higher risk of gastric ulceration, and probably due to the malfunction of dopamine in the gastrointestinal system. It modulates the gastrointestinal secretions, and it increases gastrointestinal blood flow. Right, so that relates obviously to cheetahs with their gastrointestinal inflammation, or potentially relates to them. All right, so this is my final sort of hypothesis in terms of how things function in the cheetah, and I'm going to go, because I won't be able to speak through everything. Very complex interactions that I will discuss in detail to finish off. All right, so we're eating a very low glycine diet, mainly made of muscle meat. That obviously goes to the stomach, gets digested, partially digested. We end up with a lot of these aromatic amino acids that are in the gut. That's not abnormal, that's fine, that's no problem at all. Although muscle meat contains a lot more of those aromatic amino acids than the collagen-rich tissues, in which they are normally very, very low. We get a fermentation taking place in the small intestine, bacterial overgrowth and fermentation, producing a lot of these phenolic compounds. Those have a direct effect on the gastrointestinal dopamine, as we've discussed now recently. The phenolic compounds then need to be excreted somewhere in the body. That goes through the whole glycine conjugation process. If we don't have enough glycine, we end up with sequestration of coenzyme A, and that has a massive effect on energy cycles in the body. Fatty acid metabolism, the urea cycle, the Krebs cycle, all of them are inhibited by the sequestration of coenzyme A and the fact that coenzyme A is not affected, not available for those reactions. We get, because of the low glycine, we get reduced glutathione production, which then increases the amount of inflammation from oxidative damage. We then also have reduced creatine production, which has all the effects that I've mentioned before already. So you can see that this then feeds back into the whole system, and we end up, as I said, the low glycine and low creatine feeds back into hydrochloric acid production potentially, and so that we get this whole cycle getting worse and worse and worse as the cheetah gets older. All right, I lied. I've got one more thing to tell you. All right, so this has also not been studied a great deal. There are 2 studies that really have come out of this, the benefits of gorge-and-fast feeding. So I'm going to finish off with that. So lions, you understand, in the wild will feed on a buffalo carcass or something like that, and they will probably only hunt once a week. I mean, depending on the size of the prey. The bigger the prey, the less frequently they will be hunting. They eat an enormous amount. I've actually seen them. I mean, they will consume about 15 to 20 kilograms of meat. As I said, that's an enormous amount. That stomach has to be able to expand and accommodate that. You'll see them lying with this huge belly. They will then lie under a tree for half an hour, until they've digested a very small proportion of that meal, and then they go back to the carcass and they go and fill up that other half a kilo of meat. They'll replace it and keep that stomach as huge as possible for a period of time. But then they won't eat for an entire week, okay? And you'll see the stomach going down again. Cheetahs, we know, will normally only eat for 2, maybe 3 times a week, right? And what is really, really important is the effects of that intermittent sort of feast and fast cycle on their behavior, but also on the gastrointestinal health. So in Kelsey's study, one of the PhD students that worked at Kango, she looked at the fecal consistency in cheetahs that it went from feeding 6 days a week down to 4 days a week. So in other words, going from 1 starved day to 3 starved days. And their feces improved during that period of time. So in other words, got more solid. The diarrhea disappeared, and they appeared to have improved gastrointestinal function. Okay, diarrhea would be a reflection of a maldigestion, whereas proper hard solid feces would be indication of a proper digestion. All right, and in lions, these are 2 studies that looked mainly at the behavior. The behavior of the lions improved when they were fed less frequently. All of the aggression appeared to occur over the carcass, and then there was less aggression off the carcass, in other words, the rest of the week between male lions. And they appear to be satiated. So now you actually finally got an animal that is properly satiated, whereas in between you're just feeding them small snacks, and then they never really, they're like a Labrador, never really properly satiated. All right, so it appears that by overfeeding them, they're feeding these large meals, their digestion improves, but also their behavior improves. They feel more content. So that's what's just the sort of real take-home message is besides the diet, its composition itself, it's the feeding frequency that really becomes very, very important. And we're seeing some improvements, and it's counterproductive, counterintuitive. Sometimes we think that we must feed them small meals like we do. We eat smaller meals often rather than, this intermittent feed fast cycles. Okay, but that's what they do in the wild. The only problem is I'm going to just say, is that be aware that if you have them on daily feeding cycles, you don't want to suddenly go to feeding them once a week. Okay, because the stomach capacity is not going to be able to expand to that much. You've got to kind of build them up to that amount. And then, there are lots of downsides. People like to see animals feeding. I don't know, certain zoos like to feed the animals in front of the visitors. And that can be a problem if you're only feeding them once a week. The other thing is that if you put a whole carcass there in the enclosure, visitors don't necessarily want to see a carcass being eaten, in front of them, even though that's what happens, pretty much everywhere else in the wild. So those are the challenges that we kind of need to face and get around.